So I think we're all familiar with these three sayings. They'll come up at almost any BKA meeting you ever attend. But how can it be possibly true that all of the varied practice that we use can all be genuinely working in harmony with the bees? And if the bees know best, do we provide them with enough opportunity to show us what is best for them? These questions are really important when we consider honeybee genetics. Are we really working with the bees, or are we working against them? The impact of genetics can be seen at multiple temporal and spatial resolutions. I'll start by considering some genetic consequences of the last ice age. Then I will consider the environmental conditions faced by honeybees over the last 10,000 years. These are the key ideas I wish to convey in this part of the speech. At the other end of the spectrum, incredible genetic things go on in each of your hives. I'll take you through some of the more important considerations. Firstly, the natural honeybee mating process mixes up and shares genetic material within the locality to a very extreme extent. Secondly, colony-level genetic expression works very differently to anything you were taught at school, and this really needs to be understood. Thirdly, different pieces of honeybee genetic material are very finely attuned to particular environments. Beekeepers have done a lot to mess this up. These are the messages that I would like to convey from the second part of the presentation. You'll note in that part of the presentation that some of what I describe will relate to the behavior of bees in the absence of human intervention, and that should give us some food for thought. During the last ice age, much of Europe was covered with ice, tundra, or steppe. These are inhospitable habitats for honeybees. Any honeybees in Europe would have been gradually pushed southward into comparatively small geographic territories known as refugia. Here they are, each marked with a star. Environmental conditions in each refugium would have been quite different. Honeybee colonies within a refugium could exchange genetic material through mating. However, mating between colonies in different refugia was impossible. For over 2.5 million years, honeybees in each refugium followed separate evolutionary journeys. Within each refugium, there would have been many random mutations. The vast majority of them would have been maladaptive and would have died out almost immediately. However, an occasional mutation would have provided marginal survival advantage in the environment in question. These would become established in the gene pool of that refugium and that refugium only. By the end of the last ice age, honeybees in each refugium would have become genetically distinct. Here are the same refugia superimposed on a contemporary relief map. Now, watch this. Let's start with the Apennine Peninsula. Note how it is bound to the north by the Alps. Honeybees that were forced into this peninsula at the start of the Ice Age were unable to get out again at the end of it. They became the subspecies Apis mellifera ligustica, the Italian honeybee. Next, the Balkans. This area here is bounded by the Carpathian Mountains to the north and east, the Balkan Mountains to the southeast, the Dinaric Alps to the south and the east, and the Alps to the northeast. Honeybees in this large basin became subspecies Apis mellifera carnica. Now let's have a look at this region here, just west of the Black Sea. This is divided in two by the Balkan Mountains. On either side of that mountain range, a different subspecies evolved. Lastly, the Iberian Peninsula. Something a bit different happened here. Some honeybees in the Iberian Peninsula found a route through the eastern border of the Pyrenees and colonized much of northern Europe above the aforementioned mountain ranges. At much the same time, other honeybees crossed the Straits of Gibraltar from North Africa and interbred with the bees that were already there. Thus arose the subspecies Apis mellifera mellifera, the dark European honeybee, and Apis mellifera iberica. Apis mellifera iberica is a relatively modern natural hybrid that arose from the union of two older subspecies after the last ice age. And this is very evident in the mitochondrial DNA record of the subspecies. Incidentally, there's good genetic evidence that Apis mellifera ligustica is an ancient hybrid that arose from the union of two different subspecies at the beginning of the last ice age. Each subspecies found its own route into the peninsula underneath the Alps. One followed a coastal route in from the west, the other from the east. However, the key point I'm trying to make in this section is that the 6 subspecies of honeybee that you see in this map developed in isolation from each other over millions of years. Each had its own unique genetic evolutionary journey. Now let's look at the genetic story of British honeybees. It seems most likely that Apis mellifera mellifera naturally colonized Britain by crossing the Doggerland land bridge about 10,000 years ago. For 10,000 years, the national stock would have been entirely Apis mellifera mellifera. This probably only changed in 1859, when a beekeeper in Exeter imported a small number of Italian honeybee queens into his apiary. Since then, the number of imported queen bees has increased dramatically. For example, during 2018, over 18,000 mated queen bees were imported from 16 European countries, and over 500 were imported from Argentina. Enough mated queen bees were imported in that year alone to head 1 in 14 of the colonies estimated to exist in the UK. This level of importation has been going on for many years. As the second part of this presentation will explain, imports can have a devastating negative impact on the genetic quality of a species that succeeds by genetically adapting to its local environment. I will draw the first part of this presentation to a close by comparing the environmental conditions enjoyed by British honeybees in the Bronze Age to those experienced by honeybees today. Bronze Age Britain was highly forested with very little human impact on the natural environment. Honeybees had already been established for 7,000 years. Colony density and distribution was saturated and optimized across the land, albeit in a dynamic equilibrium with diverse environmental factors. All colonies would have lived in tree cavities, of which there would have been an abundance. Every year, there would have been swarms, but every year would start and finish with pretty much the same total number of colonies. Therefore, in a typical year, roughly as many colonies would have died as there were swarms. Every single year, every colony was engaged in a competition simply to survive. Only the most suited colonies would win. Nowadays, Britain is largely deforested. Humans have made a massive negative impact on the natural environment. Almost every colony lives in a hive, any exceptions being feral escapes that die quite quickly. Total honeybee population is suboptimal, both in terms of the number and the distribution of colonies. We rehouse would-be swarms before they even issue. We feed and treat our stock at various points in the annual cycle. It is hard for unfit colonies to die if they're managed well. Any meaningful notion of annual survival of the fittest has been obliterated. Not only have we damaged the national stock with innumerable imports, the selection criteria used by some bee improvers might not always be sensible. At this stage in the presentation, I hope you can agree with these points. We will now turn to honeybee genetics as it plays out in your beehives every day and every year. Here are the things we'll be racing through, so we better get started because there's quite a lot there. Now, the queen bee originated from a fertilized egg that contained one set of chromosomes from her mother and another set of chromosomes from the sperm of a drone bee. Organisms with two sets of chromosomes are diploid. The queen performs many important jobs, but right now we will simply think of her as an egg factory. During her lifetime, she might produce 750,000 eggs, each with just one set of chromosomes. When these haploid eggs are fertilized, they become diploid again. These eggs are produced by meiosis. Before reducing the genetic material back down to one set of chromosomes, meiosis mixes genetic material between each chromosome pair. Every egg produced by meiosis is a unique combination of DNA derived from the queen's mother and father. There has been genetic recombination. Let's look at this a bit more carefully. I've circled the chromosome 1 pair in the queen bee and the chromosome 1 in the queen bee egg. In the queen bee, one of the chromosome 1s came from her mother. That's the red one marked Q. And the other one, which is blue and marked D, came from the drone. The chromosome 1 in the egg is a unique combination of code from the red and the blue chromosome. Although the eggs may be reasonably similar genetically from any queen, no two eggs will ever be exactly the same. Now, the thing about genetic recombination in the honeybee is that it is done at an incredibly fine resolution. In fact, no other terrestrial animal is known to go to such lengths to mix up maternal and paternal DNA during the manufacture of an egg. We must assume that in a natural setting where human interventions have not distorted things, it is positively good for all honeybees in a locality to mix up and exchange their DNA as much as possible. This is the exact opposite of what a bee improver tries to do. Unlike queens and workers, drones are derived from unfertilized eggs. This means that they only have one set of chromosomes. They are haploid. If there is weakness in any part of the genetic code that needs to be expressed in a living drone bee, there isn't a second set of chromosomes for it to hide behind. Any such drone will either die straight away, or if the genetic problem is fatal, or it will fail to mate when in competition against fitter rivals in a drone congregation area. Either way, the haploid state of the drone bee provides a perpetual filter for quite a lot of genetic material. We know that something like this is operational. There are 44 known visible honeybee aberrations, most of which are only seen in drone bees. They rapidly go extinct because the affected drones fail to mate. It is perfectly conceivable that many non-visible mutations also get taken out on the same basis by drone bees. Some genetic material is not expressed in the drone bee because it is only needed in a queen bee or a worker bee. Single gene mutations in these parts of the genome cannot get filtered out by drones. Certain sting deformities would fall into that category. One final thing about drones, because they are haploid, there is no meiosis when the sperm is manufactured. Therefore, every sperm made by a single drone is genetically identical. The way in which queens and drones mate can be described as competitive polyandry. The etymology of polyandry is given for you there. Now, when a virgin queen mates, she simply flies to a place up in the air called a drone congregation area. This is typically some distance from the hive, and there may be as many as 20,000 drones from over 200 colonies at this location. The queen simply flies through the drone congregation area in a receptive state, and the drones compete to mate with her. Each mating takes about 15 seconds, and about 15 drones are likely to mate with her. The queen stores sufficient sperm from these matings to fertilize eggs throughout her useful egg-laying life. With this system, queens should generally get mated quite easily. However, very many drones will never mate. Any drone from an unhealthy colony will be at a competitive disadvantage, if indeed such a colony made any drones at all. Any drone that is slow to respond to the presence of a virgin queen or is a weak flyer will not stand much of a chance either. Hence, all drones have genetically evolved into supreme athletes. Unfortunately, any drone on the wrong part of their orbital flight when the queen visits will be out of the frame too, no matter how brilliant they might be potentially as a mate. The system naturally ensures that only the very fittest drones get to mate, whilst also retaining a large proportion of chance. There is no other terrestrial mating system like this. At colony level, this system results in multiple subfamilies or patrilines of worker bees, each of which is genetically distinct. The behavior of the colony is dependent upon the combined impact of all these different subfamilies. We will look at this in more detail shortly. Now, imagine the successful mating of a virgin queen, here represented in red. The absolute ideal is for her to get mated by 15 or so local drones from neighbouring colonies in black. But not every mating flight ends in success. Some virgin queens fail to return from a mating flight. Even when mating flights are successful, if this was all that a colony did to maintain its presence in a locality, it's very obvious that the original genetic presence would shrink by 50% every successive generation. Simply to maintain the same genetic presence within the locality, drones from the same colony need to successfully mate with at least 15 virgin queens, if not rather more, for every one virgin queen of its own colony that mates. The drone is essential for disseminating genetic material of a successful parent colony around the whole locality. Here is a summary of the key points so far. Extreme mixing and sharing of the best genetic material within a locality is an obvious priority during natural honeybee mating. If different groups of worker bees have different fathers, then straightforward Mendelian inheritance and genetic thinking cannot be used to interpret the traits of a colony. It is colony-level gene expression that we will turn to now. We need to start with something much simpler than its long name suggests: quantitative trait loci. Now, when Gregor Mendel studied the laws of inheritance in the common pea, the rules he discovered only applied to 7 traits, and these are them listed here. As you can see, for each trait there were 2 and only 2 options. These are discrete traits, and they are controlled by a single gene. You are probably blissfully unaware that his laws of inheritance did not apply to any other trait whatsoever in the pea plant. No other pea plant trait is dichotomous. Instead, they are all innumerable variables that can lie absolutely anywhere along a continuous spectrum. These are quantitative traits, and all of them are controlled by the combined action of multiple genes. All I need to add is that a locus is the position on a chromosome where a particular gene is always located. Therefore, quantitative trait loci are all the genes collectively responsible for determining a particular quantitative trait. What has that got to do with beekeeping, eh? Well, imagine you are a bee improver. Here are 3 qualities that you might want to improve in your bees. Let's look at one of these qualities a bit more carefully. When I talk about a gentle colony, I am thinking of one that all of the following apply to. That single quality turns out to be multiple qualities, and some of them probably break down into smaller parts too. Is it likely that a single gene will control all of these things? Of course not. We now have scientific evidence that multiple genes are responsible for the aggressiveness or gentleness of a colony. I am sure that the deeper we explore these things, the greater the number of implicated genes will become. The same holds true for other complex traits, such as foraging behavior and hygienic behavior. This presents serious problems for bee improvers. We now understand that outstanding quality of the kind we dream of exists at the extreme end of a normal distribution curve. Only about 2% of all colonies are truly outstanding for a quality such as gentle behavior, and they essentially pop up and disappear largely by chance. No bee breeder could fix all the desired alleles between generations in a closed breeding population without seriously damaging genetic diversity, the importance of which we'll be exploring next. Just 2% of colonies considered truly outstanding for one quality, like gentle behavior, could be expected to be simultaneously truly outstanding for a second desirable quality, such as hygienic behavior. Of course, the ephemeral nature of truly outstanding colonies doesn't bother honeybees in the slightest, because the whole concept is human folly. Next, we must consider the crucial importance of intra-colony genetic diversity to the honeybee. None of us struggle with the idea that genes code for such things as the physical structure and colour of a bee. We can typically see physical evidence of different patrolines every time we open our hives. However, genes also code for complex honeybee behaviours. The very first thing a worker bee does when it emerges from its cell is to turn right round and go back in again head first to clean it. No other bee taught it to do this. Likewise, worker bees don't teach each other how to perform and interpret the waggle dance or any other behaviour essential for colony survival. Incredibly, instructions for all these behaviours are written directly into their DNA. These coded behaviours are only performed when changes in the environment trigger them into action. The thresholds inducing action are written into the DNA too. Just as there is physical variability between all the workers in your hives, so there is variability in how the behaviours and environmental thresholds are coded. This genetic variability is of incredible importance to colony health. All colonies need some workers that collect water or that seal cavities with propolis and so forth. But not every worker needs to have all these specialist skills. It's more efficient to have small subpopulations of specialist workers. Likewise, if the nest needs cooling, then it would be disastrous if the entire worker population started cooling activities at the same time. It's far better to have a phased recruitment of workers as the needs for an activity increases. The variability of genetic coding for behaviours and their thresholds helps to achieve this. This is the philosophy of Adam Smith in action. A number of research papers have shown that genetic diversity within the colony is very important for colony well-being in many fundamental ways. It is polyandry that introduces this genetic diversity. If there is insufficient genetic diversity, then the colony suffers. In fact, it is such a problem to honeybees that they have a genetic mechanism for putting inbred colonies at distinct competitive disadvantage, which I'll now explain to you. At first sight, it appears self-evident that any fertilized egg will turn into a female bee and that any unfertilized egg will turn into a male bee. However, this is not quite the case, as was described by the man who discovered why even the best brood pattern tends to have occasional empty cells in it. Those empty cells contained diploid honeybee larvae, but they were eaten rather than fed by nurse bees. We now know that if these larvae are protected from cannibalism, they will turn into diploid drones. Honeybee gender is actually determined by the complementary sex determiner gene, for which there are about 150 distinct pieces of code, or alleles. If a larva has 2 different CSD alleles, then it turns into a female bee. However, if a larva has just 1 CSD allele, then it will turn into a drone. The thing is that diploid drones get eaten before they develop. Now that's a very curious system indeed. What possible advantage could it have? Well, I think this is the advantage. It significantly compromises colonies where a queen had mated with her brother drones, potentially to the point of colony death. It compromises small isolated populations of honeybees as well. Imagine a small population of perhaps 6 honeybee colonies in an isolated location that has lost the ability to mate with any other colonies. This small population can no longer alter or increase its genetic diversity because it lost contact with the vastly wider network of colonies. Worse than that, it can lose genetic diversity over time. Any genetic material that fails to get passed on to subsequent generations during the random genetic lottery of mating is lost forever to that isolated population. As this process continues, there is likely to be a reduction in the number of CSD alleles within that same population. If you think about it carefully, you will see that the drones from these 6 colonies could only have had 12 CSD alleles to begin with. As the number falls within the population, so the chance of diploid larvae having just one CSD allele increases, and these, of course, are cannibalized. At its most extreme, 50% of diploid larvae get eaten, and this results in a shotgun brood pattern, as shown here. Now here we have pictures of a healthy brood pattern and a shotgun brood pattern of an inbred colony side by side. In nature, the inbred colony was already at a competitive disadvantage because it lacked intra-colony genetic diversity that we've already looked at. However, the CSD mechanism has surely sealed its fate. Such a colony cannot build up rapidly in spring, and it never reaches full population size. It perpetually pours considerable resource into brood that fails. It might just about survive between the seasons in the absence of more healthy competition. But as soon as a new influx of honeybees can recolonize that territory, it's essentially game over for the inbred bees. And this is how it ought to be in the ruthless natural world. Survival of the fittest also means death of the least fit. They're one and the same thing. Of course, in nature, the main purpose of this system is to weed out colonies where virgin queens have inadvertently mated with far too many of their drone brothers. However, there is another setting where shotgun brood can become a problem, and this is in the apiary of an over-enthusiastic bee improver who's kept all their inevitably small stock successfully isolated from all other bees for multiple generations. Here are the key learning points with regard to colony-level gene expression. So inbred honeybees with little intra-colony genetic diversity do not do well. However, they also do very poorly if they contain genetic material that is not suited to their geographic locality. This was first shown very clearly 50 years ago. In the 1960s, Louveau travelled all around France and examined the characteristics of the honeybees everywhere he went. He ended up describing about 20 different ecotypes, but the honeybees of Landes were the most interesting. There are 2 large nectar flows in this area every year, one in late May and the other in September. Astonishingly, he found that the local bees had 2 population peaks each year, both timed to exploit the local nectar flows to the full. This double population surge was not encountered anywhere else in France. Louveau did a little experiment. He moved a small number of hives from the outskirts of Paris to Landes and vice versa. And this is what happened. Colonies moved from the Parisian outskirts to Landes only had a single population surge, and they didn't exploit the local forage at all well. The colonies moved from Landes to the outskirts of Paris retained their double population surge, and this wasn't suited to their new environment either. In fact, they had a population surge just as they should have been preparing for winter, and they did particularly badly. Colonies from both areas were allowed to interbreed, and the subsequent generations had intermediate brood cycles that were not optimized for anywhere. Locally adapted population surges are genetically determined, and they are damaged by imported stock. The adverse impact of imported stock, even if it brings in extra genetic diversity, is a well-recognized genetic phenomenon known as outbreeding depression. Other sources have described the phenomena of local adaptation and outbreeding depression in the honeybee, most notably one of my all-time favorite bee books. But also, honeybee isozyme studies have shown that different geographic latitudes favor different enzyme variants. Genomic studies have shown that completely different alleles are favored by honeybees on different continents. And an entire issue of this journal demonstrated the benefits of local adaptation all across Europe. So when honeybee colonies contain unfavorable genetic material, can they do anything about it? Well, the surprising answer is yes, they can. It turns out that environmental circumstances often dictate whether an allele is beneficial or maladaptive to a colony. Let me show you an interesting genetic dynamic that must have been in operation now for millions of years. Transport yourself back to the Bronze Age again. AFB makes an appearance in the area. Hygienic behavior suddenly provides a survival advantage. In fact, it might be essential for survival. However, workers are diverted from their normal activities. The colony population fails to grow properly. Much energy is lost in the wasted brood, and so on. Survival comes at a heavy cost, but nonetheless, survival is what is required. But look at this. Hygienic behavior would have been a competitive disadvantage in the absence of AFB. Well, incredible as it might seem, hygienic behavior is hidden away most of the time and only really makes an appearance when it needs to. Back in the 1960s, Rothenbuehler demonstrated that hygienic behavior has a genetic basis. At that time, he understood it to be due to just two genes. Furthermore, he demonstrated that the necessary genetic traits were recessive. In essence, they were hidden away and had to be drawn out of the honeybee population. Even when fully activated, not all colonies would express them. When the AFB threat was over, the AFB-resistant traits would rapidly recede into the background again. More recently, Spivak was able to test colonies and successfully predict which ones had the potential to contribute to the development of hygienic behavior in a breeding program. Well, AFB is a brood disease, and hygienic behavior is probably quite a generic response to diverse brood diseases. Part, but not all, of the Varroa threat is brood-based. In the absence of any other interventions, hygienic behavior should place honeybees at a competitive advantage in the presence of Varroa. But it will come at a cost, as already explained. But we do intervene. We treat our bees for Varroa. Suddenly, hygienic behavior carries no survival advantage at all. Very understandably, we prevent the near total collapse of all our managed honeybee colonies by treating them. but this near catastrophic collapse of the national stock is probably what would be required for the emergence of hygienic behaviour at national level. This is just one of several examples of us interfering with very important natural selection pressure without even thinking about it. This is what can eventually happen if honeybees are not treated for varroa, although the pathway to this point would be so horrifying for us all that I can't envisage it ever being allowed to happen. The 10 blue circles on this map indicate areas in the world where relatively isolated wild or feral honeybee populations have managed to survive without human intervention in the presence of varroa. Data from 2 of these locations helps us to understand what happens. 150 colonies were contaminated with varroa and left on the island of Gotland to fend for themselves. A cataclysmic 94% of the original colonies died out in 4 years. Every swarm from the first 2 years also eventually died out. Again, this looks utterly disastrous. However, all swarms from the 4th year onwards issued from original survivor colonies, and these would appear to be faring better. There now appears to be a small but stable survivor stock. Over 40 years, Tom Seeley has periodically studied the closed population of wild honeybee colonies in Arnott Forest in the USA. Varroa struck these bees in 1992. However, there were just as many colonies in the forest in 2011 as there were in 1978. Mitochondrial and nuclear DNA analysis has revealed 3 quite amazing things. Firstly, there was a near catastrophic population collapse at one point. Just 1 or 2 colonies survived in the forest at that time. This created a detectable change in the mitochondrial DNA known as a genetic bottleneck. Secondly, some alleles that had just loitered in the background before were now expressed quite heavily, while certain others had taken the reverse journey. New selection pressure had altered which alleles were beneficial. Thirdly, that remarkable degree of genetic recombination, the incredible polyandrous mating arrangement, and those astonishing drone bees had collectively ensured that genetic diversity within the closed population had not significantly deteriorated. There is genetic evidence of massive honeybee population crashes in the distant past. Honeybee mating biology ensures that gene flow between colonies is rapid. It has been calculated that the network of colonies between which genetic material freely flows is staggeringly large. Selection pressure rapidly alters which alleles our bees express. What we don't necessarily realise is that our beekeeping practice has a very significant impact on selection pressure. Here are 2 examples, both relating to imported stock. It is positively disadvantageous for wild colonies in Italy to take a winter brood break. There's still forage around, and the ambient temperature allows bees to take advantage of it. In contrast, a winter brood break is essential in the UK because there is no meaningful forage at that time, and it's too cold to fly. UK honeybee colonies that don't take a winter brood break will die of starvation, typically in early spring, unless they are fed gallons of syrup in which case it doesn't really matter at all to the colony. Colonies that should have died will issue drones the following year, and these will spread locally non-adapted alleles in the area. Likewise, explosive colony growth and frequent swarming is perfectly suited to the conditions encountered by wild colonies in the Balkans. But it is an unnecessary nuisance for UK beekeepers. Any wild indigenous UK colony behaving in this way would have been outcompeted by less swarmy colonies hundreds or thousands of years ago and would have died out. But we perform artificial swarm manoeuvres on these colonies, and we even feed them into the bargain just to make sure they survive. Once again, colonies that ought not to have survived go on to produce drones, and the locally maladapted genetics makes its way into the local gene pool. And don't get me started on those warnings that come out every summer advising us to feed our bees because we're going to have a week or two of rain. This is a maritime climate. We've had a week or two of rain every summer forever. Locally adapted bees should be perfectly able to cope with it. If any colonies in my apiary can't cope with that sort of rain, then I'm perfectly happy for them to die. It's what should happen to them. I could go on, but I think you get the picture. So is there any hope for us? Well, yes, I think there is. Over recent years, the genetic composition of many carefully selected drones from around the world has been analysed. Using mathematics, we have developed an evolutionary map of various honeybee genetic signatures. We know which genetic signatures arose in which subspecies. We now have quite an extensive library of such information. You remember this map from near the start? We first imported alien subspecies to Britain 160 years ago. Nowadays, we import enough mated queen bees to replace the entire national stock every 14 years. We might reasonably expect the genetic composition of British honeybees to be a complete mess. But it isn't. Well, not everywhere, anyway. In Cornwall, there's a group of beekeepers who wish to conserve and restore the native British honeybee. In 2015, nuclear and mitochondrial DNA analysis was performed on samples from 23 of their colonies. Over 90% of the nuclear DNA in 14 of the 23 samples was derived from Apis mellifera mellifera, the native subspecies. Whilst there was an influx of genetic material from imported subspecies, the nuclear DNA was far from being hopelessly mixed up. Wouldn't you agree that the 14 colonies on the left of this chart look like they have always been AMM and that they have largely retained their original genetic legacy? In contrast, the 9 colonies on the right look as if they might have started off as imported subspecies that picked up quite a bit of locally adapted honeybee DNA because it was necessary for their survival. However, mitochondrial DNA analysis revealed something quite astonishing yet again. 14 of the 23 tested colonies were derived from Apis mellifera carnica colonies that had been imported at some point during the last 150 years. But which ones were they? The red ones. We need to stop and think about this for a moment. look at that one right on the left. 150 years ago, the nuclear DNA in the forerunner to this colony would have been 100% Apis mellifera carnica. Now, it is 99% Apis mellifera. This lineage has essentially changed subspecies. How has that happened? Well, genetic recombination, drone bees, polyandry, amazing gene flow, the survival advantage of local adaptation, and a little bit of help from some beekeepers. Despite all the things that we have done over the last century and a half to genetically thwart them, honeybees want locally adapted genetic material, and given half a chance, they will get it for themselves. Why will they get it? Because it is best for them. The bees know best. When it comes to honeybee genetics, we should start listening to what they are telling us. We should start working with them and not against them. When it comes to genetic fitness, this is what the bees are trying to tell us. So in summary, these are the topics we've covered in the last hour. I hope you agree that these are the main learning points. Somewhere in all that I've said, I hope there is something new for you and that you can go away and think about that in the context of these three common beekeeping sayings.
I just wanted to ask about isolated colonies. What makes a colony isolated?
Well, I think that typically the isolated colonies that we see nowadays are ones where beekeepers are trying to create a particular strain. And that's quite a common thing for some beekeepers to do. In nature, I can't imagine it would have happened very often, but the genetic apparatus is there to ensure that inbred colonies get completely clobbered.
The record number of drones to mate with a queen was 45. How many queen flights would she make? I was never sure whether it all happened on one or whether they made 4 or 5 or...
A mating flight is quite dangerous for a queen bee because she's out in the open. So ideally, if she can mate successfully and fully enough on one mating flight, then she will. If she returns to the colony and hasn't mated sufficiently, then she can make a second. And I understand there have been reported cases of 3 mating flights. But it's all observational biology. It's what that's what's happened at hives that have been watched.
Hi. Do you think AMM put British beekeepers, or would put British beekeepers, and more importantly, British bee farmers, at a disadvantage with European beekeepers who have more prolific strains like buckfast from Denmark?
I couldn't quite hear all of the question. Do I think that AMM
would put British beekeepers and British bee farmers at a disadvantage with European competitors with more prolific strains like buckfast?
I think that certainly the mixing up of different subspecies has presented problems. And the book I referred to was written when that was really reaching ahead. It's well worth reading because it's written by a generation of beekeepers who remember things before there was a big invasion of imports and what we might call hybrid bees, things like the buckfast. And the stories that they tell are definitely of damage as a result of new genetics coming in. But I think that if that slowed down or even stopped, that the bees would sort themselves out quite remarkably. I think the bits of genetics from other parts of the world that didn't really suit a maritime climate just wouldn't do well, and they would peter out, and we would get back to something similar to what we maybe once had.
What about honey productivity, though? What about honey productivity? There's
well,
no
that's
denying
fascinating, because actually, if you look at how different honeybee colonies work, then and look at their yields, then there are a number of beekeepers who try to restore or recreate native type bees. And they'll tell you that actually off one brood box, they'll have a big enough worker population, and they're frugal, and they will generate over a number of years a higher yield of honey than those that are using, say, Italians. And certainly in a good hot summer, an Italian can probably beat the pants off an AMM. But if you look at 10 years in a row and look at the average yield over the 10 years, the native honeybee will win hands down.
It sounds like it's a good idea to have lots of healthy drones, but should I stop drone culling as a varroa control?
Well, I think that there are a number of really important questions of that nature that you could pull out of what I said. And actually, I'd like to just stick with the genetics and leave you mulling over those questions rather than answering them all. But it is one of the more important questions to be asking, certainly.
One at the back. One at the back.
Hi. Obviously, the disadvantages of importing bees is fairly obvious. What about the movement and purchase of bees, we'll say, from opposite ends of the country, we'll say from Cornwall to Scotland or something along that?
Well, yes, these are very interesting questions, and there are debates about them. If you remember the paper that I shared with you from the French experience, I mean, those bees were moved about 400 miles, and they were totally useless in the different environments in both directions. Local adaptation really does seem to be that. And the thing is, when you understand how the drones work and how they connect so many different colonies together, you have your network of genetic exchange in the drones. You don't need to put a beehive in your car and drive it 200 miles. If there's stuff up there that would be useful down here, it will come through the drone network eventually, and it will sort itself out.
In spite of what you say, we're all trying to have colonies that are reasonably well behaved rather than being aggressive. However, what happens in the colony, the queen stays the same, but suddenly the colony becomes extremely aggressive. What is the genetics going on there?
Well, genetics is just one part of what dictates behaviour, and we know that sometimes the queen's pheromone production will change, and that can have a very bad impact on the unity of the colony. That wasn't quite what the question was about.
It might be worth.
Okay.
Because it's aggression, you say.
Sure, yeah. Roger just suggested that I touched upon F2 aggression. I don't know if any of you are aware of this. It's when you have a particular line or strain of bees or subspecies of bees, and after a second generation of mating, it can get very aggressive indeed. I suspect that is about how long it takes for these quantitative trait loci to get all mixed up, so that you've got a bit of quantitative trait from different evolutionary pathways, and they didn't evolve together, and you have some. That's probably how the bad behaviour starts.
You mentioned how diploid drones get eaten by the workers. How do the workers know which is a diploid drone?
Well, we don't really know, not being workers ourselves, but how that all evolved was there was a beekeeper who actually prevented the brood from being in contact with workers and fed them all himself. And they basically grew up into drones. So we know that's what they would become. How do the worker bees detect them? Well, presumably it's something pheromonal or taste-like, that they're just not right. And the workers detect that and eat them.
Okay.
I can't say more than that. All I can say is that the observation was that if those bits of brood are protected, then out pops a dwarf drone.
I've kept bees for 43 years, and they've always been my own, except for about 3 occasions when people have given me some bees. Is there anywhere where we can get our own bees genetically tested?
There are companies that will test the genetics of your bees, and I'm sure Roger would know, because Bibber has used them in the past, so you could find out about where to go for that.
Well, yeah, you can do, and I think it's around about £90 per sample. But then, of course, you've got but you know, it's expensive equipment that they're using. The problem then becomes, what really is the value of it? I mean, the situation in Cornwall was that they got a fairly big grant for it, and they did lots of samples at the same time.
Given the rate of global warming, which is increasing, are the bees going to be able to adapt as quickly to our changing climate, or are we going to have to help them as we're part of the problem in the first place?
Yeah, it's a question I often ask myself, I actually was so interested in climate change. I went back to university to study it for a while. I don't really know how to answer it nonetheless. I think that you know, if climate change really takes a grip, then we're all in for a hiding, and I think we'll all discover quite who and what survives it. But in terms of honeybee adaptability, I'm convinced that they have a very high mutagenesis rate, and there are good reasons why that should be. They're almost made in a way. The queen bee ovary is made in such a way that it's bound to have a high mutagenesis rate. And I suspect that it can adapt rather rapidly, much more rapidly than a human could. And probably more sensibly than a human could try and do. I think they'd be best left to themselves.
From your lecture, is there anything we can learn about the Asian hornet and its development?
I've never thought about that, to be honest. Honeybees and their ability to fight off pathogens and pests is another astonishing thing. I mean, it's probably the word I use the most with bees, is just how astonishing they are in every way. I mean, they've actually turned off vast chunks of their immune system. They use propolis. They do all kinds of amazing things that really make them as economic as possible. Now, you can look at the Asian hornet as an example of a generic threat, a big insect, big flying insect that comes to the front of the hive and tries to gobble up everything inside. But it won't be the first time in an evolutionary history of millions and millions of years that such a predator has ever appeared for honeybees. What has happened, which is so awful, is human beings again. We've brought 2 species together that should never have come together so dramatically and so fast. Maybe at some point there would be an interface one day in the future and it would gradually happen, but no, us humans, we bring it on, don't we? And suddenly different species that should never have come into contact with each other are. And you can't evolve a specific defence just like that. You can reveal generic defences that are hidden away, maybe, if they exist. But you can't actually expect a honeybee to suddenly come up with a load of mutations that make it, resistant to the Asian hornet. If there are genes that make them resistant to generic hive entry threats, maybe there's a bit more of a chance.
One thing that's come out of what you've been saying is, are we opening up the hives too much? Because if we're getting bees used to us opening it up every 10 days to make sure there isn't a swarm, etc., 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 from what you're saying, I think we're probably doing an awful lot of harm, aren't we?
Are we opening our hives too much? I think one of the things that probably happens as we get more experienced in beekeeping is we open up our hives less. So yes, I think that early on in our beekeeping hobbies, we're so excited and want to learn so much, want to see it all for ourselves, that we're opening up the hives all the time. And over time, I think you can learn to read what's going on around the hive entrance, and you can learn to read what's going on just from looking at a smaller section of the frames. You don't have to go through from front to back. So I think, yes, as you progress in your beekeeping, that such changes do happen to your practice, or should happen to your practice. And that's probably better for the bees.
Sorry, I know this is about honeybees, but it's interesting the comment about the Asian hornet. If you flip it around, and I actually think it might have been the question he was asking. If the Asian hornet was an isolated case in Europe of a queen bee coming in, how come that wouldn't have died out genetically? How does it? You're saying that isolated situations generally result in the demise of the colony. How come the Asian hornet hasn't died out if that's the case?
Sure. I don't know anything at all about the genetics of the Asian hornet, but you're right, there is a bit of a riddle there. And another one that I find quite a riddle, which I'm sure that if I search literature enough, there's something a bit curious about the genetics of varroa mites for me. And I don't understand how they can remain as voracious as they are. But these are not things I've looked into. I can't really give sensible answers.